Okay, it's about that time for our seminar for Wednesday afternoons here at the Bodega Marine Lab. My name is Laura Rogers Bennett, and I'm really excited to introduce our speaker for today. She is what I would call a real Bodega Marine Lab success story. She was telling me a little bit about when she was working with sea urchins here at the labs with Mark Peterson and myself as a grad student. She was an undergrad, and I remember she had to take samples every 20 minutes around the clock, so she rigged up a little bed in one of the offices here and woke up and took a sample, went back to sleep. So Andrea is one of our own. She went on after an undergraduate at Davis to get really excited about all the hands-on experience with marine research that she got here at Bodega, and went on for a master's and a PhD. She is now a professor of marine ecology and aquaculture, and the director of the Aquaculture Biotechnology Research Group. She's at Auckland University of Technology and in Auckland, New Zealand. She's going to be talking to us today about the biotechnological applications in marine science and how do we understand the language of physiology and the basic research that goes into all that understanding of how to go on and use your marine science to help with things like understanding complex questions in host pathogen interactions, microbiome host interactions, climate change resilience, and ecophysiological trade-offs throughout life history. So with that, I'd like to introduce Dr. Andrea Alfaro.
Thank you very much. Can you hear me in the back? It's great to be back. I had one of the most amazing meetings with the students at lunchtime. So thank you for those of you who were with me at lunchtime. I really appreciate all your thoughtful, insightful questions that inspire me to reflect back on my time here at Davis in the Marine Lab. Laura was saying, what she didn't mention is that I was her field assistant. So I was trying to keep up, diving behind her, doing sampling with the urchins, and I could not keep up. So I have some fond memories, and yeah, I'm on sabbatical right now until the end of the year, and it was an opportunity to revive some of the work that Laura and I have been talking about for a long time, and also make new connections. So this opportunity is great for me. And I want to share a little bit of the work that I've been doing for the last couple of decades in New Zealand. Yes, so I am the head of the Aquaculture Biotechnology Research Group. I want to talk a little bit more about this group. When I arrived in New Zealand, I realized that my rigorous fundamental research was in marine ecology wasn't going to cut it, and in order to get funding, I needed to apply that work. And the application of marine ecology to me was aquaculture. So we've been doing a lot of work in that sector, but underpinning that is a lot of very interesting, rigorous scientific discoveries and developments. So the main focus of our research is to develop and implement innovative ideas, innovative tools, innovative approaches to understand the complex interactions of organisms with one another or with the environment, and trying to tackle some of the challenging questions that are facing our environment right now, specifically climate change, resilience, and those host pathogen interactions that are presenting some major challenges, not just for the industries, but for conservation purposes as well. So the theme of our group is very much having students working within the industry, They spend a lot of time with our industry partners. We work with pretty much all the major aquaculture companies in New Zealand, as well as indigenous communities, which I'll talk a little bit more about today. So very active, very collaborative, multidisciplinary research group. This is sort of one of our current or more recent maps of the research that we undertake across many different species, not just in New Zealand, but overseas as well. And the students tend to be, and there are many different funding mechanisms to support all of this work. A lot of our research funding comes from government, but also companies that do individual contracts with us in New Zealand and overseas, big and small corporate companies, as well as community groups. And then we try to fit students within those projects wherever they feel that their niche is. And then we integrate that. And at the center of it is basically a toolbox that we've developed that has a lot of facilities, but also instruments and approaches that we use over and over and that we keep building on to apply it in different areas. So today, my first slide had mussels and abalone. I was going to talk about both, but there's just no way that I could do that. So I'm just going to focus on abalone because I know that there are quite a few of you who are interested in that from all of the work that Laura and her team and others have been involved with here. So I'm going to try to focus just on abalone, and I'm going to give you some very brief highlights of some of the work that we've been doing in different projects. So I'm just going to weave in from one project to another. So if you get lost, just try to follow through and we'll catch up at some point. But the gist of it, I think you'll get. So abalone is a very important species in New Zealand. We have 3 main species, but the one that we focus on commercially is called, well, they're all called paua, which is the indigenous name for abalone in New Zealand. So if you say abalone to a Kiwi, they'll probably go like, what? What are you talking about? Oh, you mean paua? Oh yeah, we need to get a feed of paua. So Haliotos iris is a beautiful abalone, and it's just gone in. I borrowed that one from Laura, so you can see what it looks like and how it differs from the many species that you have here. So as I said before, we focus on academic knowledge generation, but we also use traditional practices and knowledge, which is very much embedded in the fabric of research and day-to-day life in New Zealand. It's a bicultural country, bilingual, representation of Māori in government and in every aspect of the country. So all of our research also has to be embedded in that context, which provides another lens and flavor and I believe an enrichment to the research that we do. And then growing industry, so we have a very strong fisheries for abalone, but also a small growing aquaculture industry. So and also abalone in terms of managing conservation practices, especially in these Māori communities who have customary regions that they manage themselves. So yeah, so I've already mentioned that our research is embedded into Māori culture and traditions, and for that we also need to follow protocols of understanding, engagement, respect, and learning from one another. So most of our projects are co-developed, so with partners in that space. With that in mind, abalone especially is a taonga species, which means it's a sacred species for Māori. So there is a sense of connection that goes back a long time in the sort of oral traditions. I don't know why it's changing for me, but anyway, some of the sort of traditional stories that Māori tell are Tangaroa is the god of the seas, and when he went down to look at the paua, he saw that it was a sensitive species, and so he said, oh, I'm going to help you, and I will take from my domain the coolest blues of the oceans and ask of my brother Tane, who's the god of the forest, to give you the greens, and from the dawn you shall have a tinge of violet, and from the sunset a blush of pink, and overall there will be a shimmer of mother of pearl. I think that kind of sets into context a lot of the things that we talk about, because Tane, the god of the forest that provides the green, of course, has an ecological connectivity from the forest and the nutrients that come down from the mountains in the catchment through the rivers that then in the traditions of Maori culture is the breath of life that comes from the mountains, comes through the rivers to feed the kelp, and the kelp become big and strong and powerful, and then the power comes in and steals that breath by eating it, and then becoming growing, and then when we eat the power, we get all of that connection, that breath of life from the mountains to the sea. If you're an ecologist, you can talk about nutrients coming from the catchment, feeding the seaweed, which then grows, and then of course grazers come in and eat that. So it's about thinking in different ways about the same thing, really. Yeah, so we work with communities, we have a lot of engagement, and they're part of the framework. This is a marae, so a meeting house where all of the initial conversations about any projects happen, all of the protocol. Once you've established that protocol and you start developing the trust, then the students can come in and start doing their project if they're allowed to be working there. Okay, so moving on, so I'm gonna come back to that traditional knowledge later on, but I want to sidestep now and share with you a little bit of the research that we've been doing with fisheries. I mentioned that fisheries is an important aspect of seafood production in New Zealand. Is the majority of the seafood, of the paua, goes to overseas markets, so it is economically important. But marine heat waves have been affecting these fisheries. The divers are noticing big die-offs, so they were really concerned about that. A lot of these fisheries are managed, most of it is managed by Maori, so they wanted to look at this mortality. They also were looking at stunted populations as being particularly important for us to address, as well as what they thought were big disease that were causing big mortalities and biomass suppression. So they wanted us to look at this problem. And of course, we applied our multidisciplinary approach to investigate these problems. So I'm going to give you just one example of some populations in the Chatham Islands that we were privileged enough to study. That's where 30% of the abalone fisheries comes from. And this location is pretty much off the east side of New Zealand, about 700 kilometers, whatever that is in miles. I forgot the conversions now. I don't think about miles anymore. Anyway, you can only get there by, well, we fly there with all of our gear. And what we did is we looked at some areas. So these are 4 sites: fast-growing sites and stunted populations. So stunted populations are populations where the adults never get bigger than a certain range, so they are not able to be fished, and so they wanted to know why this was happening, and we did a multi-pronged study in that area. So that's just to give you a sense of where. So these are some of the divers that were helping us in the area. It's very isolated, so the paua here are literally in the tide pools, like massive animals this size. You have to walk very carefully not to step on them. So we could easily go and harvest them without. So this is pretty much how it is all over the place. They're just covered with large abalone. These are some of my students just harvesting in the intertidal zone, just to show you where the site is. Okay, so our approach was pretty much doing some morphometrics, understanding what the habitats were like, and for that we did some surveys where one of my colleagues has developed this really cool method where he flies a drone and then connects, and then we have a diver with GoPro cameras and a transect line. The drone goes above and they're connected. So when you've got the video showing you, I don't know, Olva lactuca, a particular seaweed species, you know what it is and connects to the drone, and it can map that particular wavelength so that you can actually understand the distribution of species as well as the abundance and diversity. So we did a very big habitat analysis and then comparing the sites. We also have developed a flow cytometer that is this big and comes to the field, so we can do some blood analysis on site, and then there's a whole bunch of measurements, immunological measurements like total antioxidant capacity and so on that we can do with that. We've obviously collected some samples for histology, and then metabolomics is an approach that we actually were the first ones to implement in aquaculture research, and it's been super successful for us. We have a really nice work plan. In this case, we used muscle tissue and hemolymph to analyze the signatures, and I'll talk a little bit more about that in a minute. We did some gut content analysis and microbiome to understand what was going on, because we thought there were some diseases that were impacting these populations, but we'll find out in a minute what actually happened. So just in a nutshell, there were clearly lots more biodiversity and abundance of seaweeds in the fast-growing sites. There was a lot more bare ground and sediment in the areas where the stunted populations were. The morphometrics gave us that information as well. When we looked at the histology, we didn't really find a lot of parasites or pathogens, which was indication that that maybe there wasn't a disease issue, but there were definitely differences in gut content. So the fast-growing populations have very nice fresh seaweed fragments compared to the stunted populations that had degraded algal pieces or lots of sediment. And then the other thing we found is healthy versus unhealthy digestive tubules. So in the stunted populations, there was the digestive tubules were separating from the lining, which is an indication of unhealthy population. The gut content analysis corroborated that with clear fragments of seaweed in both the juveniles and the adults compared to the stunted population that had a lot of sediment in them. So we started to get the picture that there was some nutritional thing going on in here. And when we looked at the flow cytometry, again, these are some of the types of analysis we can do. Obviously, blood cell count and viability, oxidative stress via ROS production, apoptosis or cell death, and among others. There are many other tools that we've developed with this machine. By the way, we borrowed this machine from human blood analysis. So the Roche lab uses this for human blood, and we went and took it and thought, I wonder if we could do this with invertebrate blood, and it worked out. We developed the protocols. Now we can get a sample in 2 minutes. We've got a result for all the profiles. So it's fantastic, and because it's so tiny, we could take it in the field and do all of these analysis in the field. So what these results show really is that there were no significant differences between any of in terms of cell, I'm just showing cell count and viability here, but there were no significant differences between the sites. So there were no major signs of immunocompromised state or unhealthy animals. So it was pointing that it wasn't a health issue, but more of a nutritional issue. Now we use omics, the omics cascade a lot, especially in metabolomics, but I guess the idea here if you don't know, is that we go from the genome or the genotype to the phenotype, with genomics being the whole study of the genome. And I like to think about the fact that the information level here is about what can happen. It gives us information about what can happen in a biological sample, whereas transcriptomics tells us about what appears to be happening, and then proteomics is about what makes it happen, so the proteins that are involved. And then metabolomics is really about telling us what is actually happening or has just happened in that organism. So it's much closer to the phenotype, information about what the animal is sensing from the environment, whether it's pathogens, thermal stress, ocean acidification. It's actually giving you the signature of how the animal is experiencing the surroundings. So it's very, very informative. It's, to me, it's almost like the animal is telling us what's wrong with it, or what's right, or is it happy or not. And so it's a very, very powerful tool to understand what's happening to the animal as we are seeing it right now. Very, very sensitive. And then we use omics integration to put it all together wherever it's possible. So a little bit more about metabolomics, because this is one of my passions, and I really like working with metabolites. They're very conserved across biological tissue. So it's really about studying the metabolite profiles and signatures. So metabolites are really small molecules that are found in every biological sample, whether it's cells, tissues, biofluids, or entire organisms. We call that the metabolome. There are downstream products of gene expression, so after translation, so they're very, very sensitive to environmental signatures or changes, as well as, pathogens. They're good descriptors of the organism's phenotype. And then we can study them to understand biochemical pathways that are activated within the organism as a response to interactions with its environment, and so on. So our metabolomics approach is basically, you can do targeted or untargeted. Most of the times we start with untargeted metabolomics, which is basically study the whole profile and then start to pick out what's interesting, and then from there you generate hypotheses that then you can go and test with targeted approaches. So mostly what we do is the analysis of metabolite profiles of whatever sample, and the way we do that is we collect samples from a tissue, freeze dry it, put it in liquid nitrogen, freeze it right away, capture that signature so that we can then analyze it in the lab. But we need to capture that signature very quickly, because as soon as you start manipulating the animal, you introduce more bias, more stresses, and therefore more changes in the biochemical pathways that cover or that interfere with your signatures. So we use this approach then to identify biomarkers of a specific condition that we might be interested in, and then we superimpose those signatures on biochemical pathways and start to look for associations. It's a deep sort of mining of the data and trying to understand and describe these biochemical pathways and how those reactions change with a particular pathogen or environment, and so on. So this is just as an example of that study that I was just showing you, where we had hemolymph and muscle samples, and then we can start to tease apart metabolites that are up or down regulated as a response to, and then and responsible for that difference between, in this case, the populations, the stunted and fast-growing sites, or the life stages, adults and juveniles. And you can see that different tissues give you different signatures, because different things, different biology is happening in different tissues. So if you're talking about respiration, collecting the gills might be a more important tissue for you to collect. If you're talking about reproduction, maybe the gonad is more important for you to collect. And so we've done a lot of work in trying to understand how different tissues differ in terms of their signature. So we see, clear differences between the fast-growing population and the stunted population, showing that there was a range of variability in the stunted population. That condition has potentially different outcomes. And when we look at the two different groups in this heat map, very, very big differences in a whole range of biomarker metabolites. And then from there, we start picking, okay, which ones are more indicative of that particular stress condition. So when we find these very important, potential biomarkers, we start putting those over known biochemical pathways, and we start to understand whether there are any major disruptions. In this case, there were some major energy pathway disruptions that were suggestive of catabolism. So the animals basically starving in the stunted population, starving itself, and again supporting the fact that they didn't really have enough food, and that while they weren't dying, that long-term period of malnutrition was causing the stunted form. In the fast-growing population, we have a lot of energy that is being used up for growth, and so the whole story started to come together and in a consistent finding. So in conclusion for this study, we found, yes, that the two populations had significantly different habitats, with fast-growing abalone having a lot more seaweed diversity and quantity to feed on, whereas the stunted population was more starved, and it's a long-term starvation that was causing that stunting. We've confirmed that in other sites ever since then. So it was not an immunocompromised situation. And then the other thing is that the juveniles in the stunted habitat were still able to find enough food to survive, but it's just a long-term, it wasn't enough to grow to a big size. So long-term food limitations were the issue. Then we went on to look at the marine heat wave factor, and for that we collected, as climate change is affecting the oceans very significantly. Temperatures increasing. There's plenty of evidence to show that. We've got records showing massive increase in temperatures every year. I think at the southern point in New Zealand, we've got 2 degrees warmer this year than last year overall. So there's definitely an impact of warming temperatures. And these marine heat waves that come in are just causing tipping points for these populations. So we wanted to look at what was happening. So we collected animals, again, from many sites. This is just showing 2 sites, again with hemolymph and muscle, to try to understand in the one stunted and one fast-growing population what was happening with the abalone that were collected at different times. So we tried to go to collect samples before, during, and after a marine heat wave. It's not always possible with divers. They don't want to go when it's rough conditions. I don't know why. Anyway, but we did get some comparative dates, and then we applied our multi-technique approach to understand this. And just to give you some highlights, because I realized that I can't go into too much detail. In terms of histology, we found that the animals were achieving reproductive, they were ripening the gonads during the warmer temperatures, but there were some different outcomes. And the main problem was atresia, which is basically the reabsorption of gametes from an animal when it's finding a stressful situation, and the reallocation of that energy for other survival type systems. So The fast-growing population was holding on to these oocytes and over the spawning season and wasn't actually producing, they weren't spawning. They were developing, but they weren't spawning. The slow-growing population, some of them spawned completely and just reabsorbed. So that's Atresia. And so they were basically not producing enough gametes or gametes for the next generation. So a significant worrisome reproductive suppression of these populations. We've since gone and looked at this in other parts. Even in the south part of New Zealand, we're still finding populations that haven't spawned for years. And in the farms, we get broodstock that are not spawning because the water is warming in those areas. We're looking at the whole east coast of Australia. We have a project with University of Tasmania. The same thing is we're finding there. Talking with Laura, she's got the same sort of worrisome worries for here. When we looked at the pathogens, the stunted population was more susceptible to having these pathogens. So again, it's a compounded effect of thermal stress that weakens the animal, and then it's more susceptible to pathogens. So in summary, for that section, we've got stunted populations having low quality and quantity of the seaweeds in their guts, and then there are, I forgot to mention, there were more ceroid. Again, this is a signature for stress, as well as kidney stones in the stunted population. Reproduction suppression, so number of ripe gametes, they were there, but there's very strong indication of atresia. And then parasites were increasing in prevalence in the stunted population. We looked at the microbiome, the within group diversity. We definitely saw a decrease. So during the marine heat wave, at the peak of the marine heat waves, we had a decrease in richness and diversity. When you look at that in more detail, this is just beta diversity comparisons, very distinct differences, and we started looking more specifically at what these communities were. I have a student who's taken a very long time to try to tease this apart. There are some very good signatures of stress communities and dominance and relationships that are giving us information about functionality. So how the stress response creates a community that has a particular function that affects the metabolic processes in the host that are consistent. So if you stress the animal, thermally stress the animal, that community changes in that particular, or that functionality of that community changes in that particular manner. We don't know exactly which are the microbes that are involved in that. That's a lot more difficult, as we were talking earlier today, to tease out. But yeah, so that's an ongoing bit of work, as I said. But in summary, richness and diversity parameters were lower in the stunted population and also during that marine heat wave peak, and then some dominance of key bacteria associated with that temperature signature. When we look at the metabolomics for that, I've shown you some of these analyses, but clearly the population that was collected during that marine heat wave, very different from the pre and post heat wave population. When you look at that, then we tried to superimpose that in different biochemical pathways. This is just central carbon metabolism for both hemolymph and muscles. And so we try to map this over, we look at what's up and down regulated and whether there are things that come up all the time with that heat stress. I'm not going to go into that. I can talk to you more about that, but these are lipid metabolite responses for thermal stress, and these are just amino acid metabolite responses, again, for thermal stress. So it's like an investigator just trying to find out how these metabolite signatures are affecting the physiology of the organism, and then finding markers for that particular condition that then you can use to assess your population. So yes, they might be dying, but you don't know of what, but you can just take a blood sample and look at these metabolites. If you find those biomarkers, you're like, oh, there's a pathogen going on here, or thermal stress, or nutrition, or whatnot. Yeah, so this is just pre-marine heat wave, during marine heat wave, and post-marine heat wave in a heat map of certain key metabolites. And you can see clear differences between the two, but then again, like the other picture showed you, significant changes in metabolites that are affected by thermal stress. And in conclusion, you can see that there was an increase in some metabolites associated with energy metabolism. It's the signature for stress, thermal stress, sugar-based molecules supporting glycolysis, functioning, again, ATP generation, as well as amino acids that feed into the CCA cycle, and then biomarkers of that, just a simple one, lactate, well-known indicator of anaerobic stress, energy metabolism, and also we associate it with thermal stress, rapid, shifting that energy to other sources. And the other interesting thing we found here is nucleotides that were also affected, suggesting increase in RNA and DNA turnover, which are well known to be associated with potential cell damage. And this is a really interesting area because we've now used transcriptomics to try to understand which of these potential gene expressions we can start to manipulate to maybe create a more resilient juveniles for the next generation in a farm condition, for example. So this is, again, ongoing studies. We have now very, very good signatures for thermal stress and specifically marine heat wave events. We know metabolic pathways that are affected and in what way, and and then we started looking at these reproductive suppression signatures, and now we've got, as of recently, we've got some signatures that are starting to show promise into giving us an indication of reproductive suppression without, and the application of that, of course, is very clear that if you've got a selective breeding program and the animals are not spawning, you can actually use this method to save time trying to spawn animals that are never going to spawn at that time. Okay, shifting gears a little bit. I'm looking at the time. Abalone, this one is in aquaculture setting. So this is feeding nutrition. So in New Zealand, as I said before, there's a growing aquaculture industry for abalone. All of their farms use formulated feeds. There's a need for species-specific and season-specific feeds to increase the growth and immune system and so on. The feed cost is about 50% of your production, so there's a clear need to reduce waste, and there's a lot of waste and contamination of the water. So we've developed a really cool encapsulated feed that has the ability to add resilience and sustainability to the industry. And we've been lucky enough to work really closely with an abalone farm that's been ideal. They just are our playground, and lots of students work there. And so we started trying, we've had a few students do a thesis on this topic. This is one of the more interesting theses that just finished not that long ago, where we use alternative feeds for a more sustainable production. So we use insect meal as a substitute for fish meal, this is just a low value byproduct. It's very easy to grow. You can grow soldier flies and other insects very easily. And then grape mark, which is a byproduct from the winery, the skins and so on of the wine. There's a lot of antioxidants and fiber in there that we can use. And we can we've been formulating different feeds. Some of them we've shipped here to the lab for testing. Dan Sweezy's got the feed in his lab now. And the idea is to, and we've developed this encapsulation that's got a really nice polymer, and we've added some compounds to the polymer that, to our surprise, they just love it. It's 100% consumption. There's almost 0%, because the stability is so good, there's hardly any leaching, so there's no pollution of the water, and it stays and its integrity up to 11 days. So one of the farms that we used this or applied this to, they were able to go from feeding every day to feeding every 2 days, which saved a lot of money, but also having to clean less, and the animals ate everything. And we are, with a probiotic that we developed for them, now they have a 25% increase in growth just on that one. The best that we've been able to increase growth with some of the newer feeds is 85% growth increase, which is huge for an abalone that takes 3 to 5 years to get to commercial size. This is what the feed looks like when they're trying to eat it. So it's a little ball, and I just can't get over the fact that they just love this, and they grab it, and it's almost like chipmunks of the sea. They hoard them, and then they just, when they're ready, they just slit one into their mouth, and yeah, it's really cool. Unfortunately, the video is not the greatest, but I think you can see the activity of how they I've been trying to get a student to work on just describing this behavior with different shapes, but I don't know, they're not motivated yet. Anyway, the methodology for this work was, just as an example, we've done quite a few of these, trying different diets. This one had inclusion of insects and grape. And again, we use our whole toolbox to try to understand from different points of view what's happening with the animal when we manipulate, in this case, the feed. So of course we can look at proximate analysis, and we can look at amino acid profiles and fatty acid profiles to see the improvements on the feed and which ones are showing us more benefits. And the cool thing is that we can actually do lots of, we can gain a lot of information like taste, texture, color, all of these attributes that the industry is quite interested in. From a marketing point of view, we can actually work on trying to improve those, and we've been very successful with some pretty innovative projects in that space. So as a summary, our integrated omics approach really takes a look at a whole bunch of range of aspects that affect the organism. So maybe pathogens, environmental stress, or toxins, or developmental issues, growth, and so on, whatever the industry is interested in. And then we apply the multi-omics cascade to wherever is most appropriate. My bias really is metabolomics has been such a successful tool for us. But then sometimes when we're trying to manipulate the genes or the genetic expression, then we can go back to other omics and integrate that information. This is just one example of integrating metabolomics with proteomics, and this was another study where in Australia, where the fishermen were collecting the abalone and taking them to holding tanks, and they were going to be shipped to China, and then COVID hit, and they were holding these animals forever, trying to feed them in the tanks, and of course the water was getting polluted, they were dying, so they wanted to figure out, can we get a signature, or can we get a feed that doesn't pollute? And so we did the same thing. We sent some encapsulated feed, and we tested. With this approach, we tested before when, post-harvest, or just as they were harvesting, holding tank, and then after the feeding. And the signatures with the encapsulated feed reveal that the profiles or the physiology of the animal went back to almost exactly the same after the feeding as when they had just harvested. So with that feed, they could keep the animals for longer in these holding tanks until the China markets opened again and they could actually send them offshore. So we now have very good biomarkers of nutritional, of nutrition and a nutritional boost that also helped us reduce the pathogen load. So when these animals get to Chinese market and they like to put them in these tanks for the viewers to pick, they were saying that these animals were lasting a lot longer and the pathogens that commonly affect them there weren't so much of a problem anymore. Yes, now we're looking at, we've got students working on seaweed inclusion. So to conclude, our encapsulated feeds are, they've been great for commercial purposes, more sustainable, less pollution. They increase growth rate by a whole lot, and now we're showing flavor and tenderizing benefits as well. Gonna run out of time. Okay, back to the community work, looking at the paua and its ecosystem. I said that it's a Tonga species. It's also a tohu species, which means it's an indicator of environmental health. So we can use this information to work closely with Maori communities, its relationship with the rimu is the seaweeds, and then that connectivity to the rivers and catchments. And I mentioned that it's believed that the life force comes from the mountains and through that cascade back to the human population. So when you feed, when traditionally they eat, paua is not just for nutrition, but it's a life force enhancing, and community enhancing aspect. Another really, and that is shown by the fact that paua has a human, it's a living thing that is connected to humans. So for Maori, the paua is a cousin, is part of the family. So when you're eating the paua, you have to respect that the animal's making a sacrifice to feed you. So, and it's giving you life force. So the carvers, when they carve their the ancestors, these are ancestors that adorn the maraes, meeting houses, and so on. The last thing they do is they put paua shells in the eyes of these ancestors. And as soon as you do that, you bring life into the carving. So those are live entities, so they feel connected to the ancestors when they go into these meeting houses. So it's quite important that we appreciate that. And so with some of my Maori students, we've been doing a lot of work in trying to weave in Western science methodology and traditional knowledge and applications. And when you do that, you find that there's immense parallels that are really interesting. So for Maori, when you're in the present, you look to the past to move to the future. And if you can integrate what you've learned in the past, where you are, what you've learned in the past, and where you're going, you can actually arrive to an ideal state. And that ideal state has to do with the health of the environment first, and then with that, you've got healthy people in that environment. So it's a different way of thinking from what many of us are used to. But also, we have the scientific method, that we teach and we learn in classes, but Maori believe in the observable state of the balance of the ecosystem, our understanding of that state, and then the actions that you must do as kaitiaki, or guardians of the environment, to ensure that that sustainability of the ecosystem persists for the next generation. So these colored words are Māori guiding principles. And when you superimpose so for example, genealogy, whakapapa means genealogy, so where you come from is really important in Māori, and you're not just you are where you come from. That genealogy is really the genetics of the Paua, which is also represented in the genetic of ourselves. So by understanding that genetics, we understand the whakapapa, where we've come from. So that's just one example, but weaving this concept, you can actually understand and have a more comprehensive view of the environment. And working with Māori communities, what they've actually signaled is we can't deal with climate change. It's something that our ancestors didn't experience. We don't know how to deal with that. We want to actually engage with Western science in a culturally safe manner to actually help us be better kaitiaki, or guardians of the environment. So we use our toolbox, which goes from multi-generational potential resilience to transient resilience. So where can we add resilience? So whether it's the genetics or selective breeding programs to offspring provisioning, carryover effects, maternal investment, all the way to nutritional boosting through diets and interactions with key microbiomes to add resilience at the whole, everywhere that we can to produce an abalone offspring that are more resilient for future climate change. That's just an example. We've been able to find some bioactives in seaweeds that are commonly eaten by Paua, and we can grow the seaweeds in a particular condition to enhance those bioactives that then we can use in the encapsulation technology to feed our captive Paua, and therefore produce the next generation of Paua that can be put into these rohe, these managed areas that are managed by Māori, and they can put them inside these cages and use traditional methods of clipping seaweed to feed the Paua, which only needs to be done once a month, as well as working with companies that are focusing on cultivating, farming these seaweeds to again feed the Paua. And this is a project that we've been working for a little bit and hoping to get more funding to do this work. These are some photos from one of our colleagues in France, but this is the technology that we're trying to use. These cages, they get full of seaweed that you can clip and put back in the cage, and the Paua inside the cages that are feeding, and then when they get to a particular size, you can just open, because they're in the same environment, they're going to be released. You can open the cages and they just walk out. We're working with modelers, fisheries modelers, to help us understand how can we balance the ecosystem so that, and Māori traditional knowledge to understand, the kina is the urchin populations are exploding, so we need to harvest them and have a big feast because they're competing for the seaweed. So with the modelers, we can actually get an understanding of how to manage the ecosystem, the entirety of the ecosystem, and have the Māori themselves look at how they clip seaweed, if they need more cages, whatever. And working with these companies that are cultivating seaweeds in the lab and putting them out to farm as well. We have really good hatcheries now that are starting to be replicated, and what we need to do is add the resilience piece as well as the encapsulated piece. I'm going to finish, I swear. And just to highlight that in New Zealand, we've got universities and institutes that do research, but we really have to partner with the counterpart, which is the marae, where the traditional knowledge is shared in a safe environment, and our students can go back and forth, getting the best of both environments and teachings to apply a comprehensive inclusive framework to study the environment and its conservation, as well as the future health of human beings and everything else that's alive in the planet. So thank you very much, and I'm sorry that I went over, but thank you.
Wonderful. Well, thank you, Andrea. And I'm sure we've got a few questions here in the audience. And then if we've got any questions on the chat, please write your questions in there and we will ask after the in-person audience. So let me pass around some microphones. Please pass that down. I'm really appreciating the way you've really taken the aquaculture setting and used that as your laboratory. It's just wonderful. Go ahead.
Hello, Dr. Alfaro. I was just wondering, when you were harvesting and looking at those metabolites, how did you differentiate between those like heat shock proteins and like the added stress metabolites when you were like looking at your data from the heat wave?
Yeah, so heat shock proteins, usually you use transcriptomics. The metabolomics are clear signatures, so you just have to look at the metabolites. And I think what you're asking, I'm not sure if I understand your question, but usually what we do is if you have an untargeted approach, you don't know what's happening, so you just look for biomarkers or signatures that are odd, and then you can actually test those hypotheses. If you use a targeted approach, then you would have an experiment where you increase the temperature here, lower it here, and then you keep everything the same, and then you are just testing for temperature. And so whatever metabolites are up or down regulated are probably indicative of that thermal stress. Does that answer your question?
Yeah, thank you so much.
Of course.
Any other questions?
Thank you so much for your talk. I am not a marine biologist, so this might be a bad question. Maybe hopefully the abalone people won't laugh at me for this, but you mentioned, I think, the effect of probiotics on the abalone's growth, and that was something I thought that was kind of a nascent science, even like mammals. So I was curious how you thought of that and what the effect of those probiotics were in the abalone specifically.
Yeah, there's been a lot of work on probiotics for humans and for animals. So probiotics tend to be species specific, even season specific. So the way we found those probiotics is basically going to the farm, picking some paua that were fast growing, doing some swabs from different parts, gut or whatever, and then culturing the bacteria to see what we got. And then out of whatever, 13 or so, then we tested those. There's standard protocols to test for ability to hydrolyze. Well, there's a whole bunch of things that you test, and then you end up with some indicative potential probiotics. And in this case, we had 3 bacterial strands that were producing good results. We included all 3, and then we tested that with a growth experiment, and then we challenged the animal, and we saw improved immune system as well as growth. And so then that's how you develop a probiotics. And then once you've got a reliable probiotic, then you can cultivate it in the lab, bioreactor, and then you can dry it and include it in the feed. There you go.
Any other questions?
I just had a question about the biomarkers that you mentioned, studying the marine heat waves. I guess I was a little curious whether you were characterizing it by like the aggregation of the metabolites or whether it was like an expression thing. I think I just got kind of lost there.
Yeah.
And
Thank you.
what that represented.
Sure. Thank you, Jordan. I think there are, when we talk about biosignatures, they can be a lot of things. One of them could be a metabolite that is upregulated, or its abundance is much higher than you would expect it, and it's signaling maybe a biochemical pathway that's activated to produce that metabolite. And so because we're picking that signature, we know that there's got to be something going on in that pathway. Other times it's a combination of metabolites. Other times absence or reduced amounts of that metabolite. For example, itaconic acid is one of the amazing probiotics that we found for bacterial infections, and we've done a lot of publications on that. That one's got some evolutionary implications because it's in an innate immunity system, and it's only previously been found in adaptive immunity. I'm going off track, sorry. Back to your question. Your question was about how do you identify a biomarker? Is that right? Yeah, so when we find something that it could be a potential, then we do targeted experiments until we find whether thermal stress produces that one metabolite every time. And then that gives us an indication that that could be a good biomarker. So
Okay, thank you.
not just abundance, but sometimes relative abundance or combinations of.
We have one question here in the chat. Is there any evidence for slow growth in New Zealand paua promoting resilience to environmental stressors?
That's a really good question. Not that I know. No, but it would be really good to look at that more closely.
Yeah. Because what you're finding is more that it's a lack of food rather than a genetic adaptation
Yeah.
that might have some additional
And
benefits.
in the farm, we find, the only thing I can think of, in the farm, we find animals that have not spawned forever in the broodstock, and they're not, doing very much. They're not eating very much. They're great, but when there's a high increase in temperature, the ones that are happy to move around, so potentially.
Interesting.
I have to think about that. Whoever asked that, email me.
Great.
Very interesting.
Well, I think we'll give Andrea one more applause, and please join us after the seminar in the lounge for the 4 to 5 o'clock relaxation hour. Okay. Thank you.
Thank you. Thank you.